Ray, do you want to wait just another minute or so to get started?
Yeah, that sounds good.
Okay. Okay, we're probably at steady state.
Cool. Great.
So my name is Patrick Kinnunen, and I'm a fourth-year PhD student in the chemical engineering department. I'm in Dr. Jennifer Linderman's lab, and I'm also very closely advised by Drs. Gary and Kathy Luker. Ray, do you want to introduce yourself and get us started?
Yeah, so my name is Raymond Asare. I'm a third-year PhD student, same situation as Patrick, advised by Dr. Jennifer Linderman in the chemical engineering department, but we work really closely with Dr. Gary and Dr. Kathy Luker in the Department of Radiology. And I think it's safe to say that we're really excited to talk to you today about some of the collaborative work that we've been doing between both of these labs, focusing on experimental and modeling techniques to understand cell signaling.
One thing before we get started, if you have any questions, since there's two of us, we can monitor the chat. And so if you have any clarifying questions, please feel free to type them in the chat throughout the talk, and there'll be plenty of time for questions at the end as well.
Yeah, absolutely. So I want to broadly talk about heterogeneity in the context of cancer because cancer is a heterogeneous disease. So tumors are composed of various cell types and environmental structures that create variations in the tumor microenvironment. So in this cartoon on the right, I'm showing you some of these tumor features that they consist not only of these cancer cells that I've highlighted in blue, but also stromal cells in yellow and immune cells and vasculature that I've outlined in red. And we're curious about how all of these different components interact with each other, but we're really specifically interested in understanding cancer cell behaviors. But you'll notice that even if we look just at the cancer cells, which we're interested in, they're not homogeneous in and of themselves. So in this schematic, although all of the cancer cells are blue, genetic differences and things like that really give these cancer cells different properties. So we can think about these variations under a couple of different buckets: genetic differences between different types of cancers, epigenetic differences in gene expression, and also environmental differences that alter the way a cell is poised to function. So instead of having a cluster of cancer cells that look like this, you can imagine a group of cells that looks more like this, with all sorts of different behaviors depending on the genetic and environmental inputs. So just to make this really clear, the diversity that we're seeing in these cancer cells comes from genetically inherited variations and also across environmental plasticity. So for example, if we follow the trajectory of one of these blue cancer cells, we can see an example where it divides into two cells, one of which is really similar to the parent in blueness, and the other which is incredibly different and red. And this could be a genetic or epigenetic difference in the regulation of gene expression. But we can also look at variations from environmental plasticity. So let's suppose that both of these cancer cells are all of a sudden placed next to a stromal cell. We might see the blue cell shift from a blue to a black color phenotype, while the red cell is, for whatever reason, not disposed to change in the presence of this neighbor. And furthermore, we can think about different chemicals or biochemical inputs in the cell environment. So this could be a drug or a chemokine, which we're really particularly interested in, secreted by different cells in the tumor microenvironment. And in the presence of one of these chemokines, this black cell might die and not be able to contribute to the progression of the tumor, but the red one could go on to continue and survive and proliferate. And so here, hopefully you're beginning to see just how difficult cancer treatment is in these heterogeneous populations of cells because therapies don't affect all of these cells equally. So small subpopulations of cancer cells like this red one are the ones that really drive disease progression. So you can think about drug resistance, or this red cell being the one that's able to escape the primary tumor, survive in the immune system, and then colonize a new metastatic site to make cancer lethal. So it's important for us to understand how these single cells and rare populations make decisions that promote cancer survival. So one of the ways that cancer cells do this, that our lab studies, is the way that they respond to gradients of chemicals like CXCL12, which are present in the tumor microenvironment. So here I want to introduce the concept of receptor-mediated cell signaling, which is how cells take in information about their environment through receptors on their surface and convert that information into behavioral responses like promoting migration. So in this case, you can see a gradient of CXCL12 in yellow that binds to this cognate ligand CXCR4 on the red cell surface, which ultimately leads to downstream oncogenic behaviors that we want to understand and control. But this process to control behavioral responses is actually really complicated. And just because one ligand binds to a receptor, it doesn't mean that we'll see the same oncogenic behavior happen in different groups of cells. So that gets us into different cell signaling behaviors. So there are numerous signaling pathways inside of the cell, and phosphorylation and different modifications that go on to activate specific signaling pathways. And two of these pathways that we study are AKT and ERK, which are turned on by kinase activation downstream of the receptor ligand binding event. And these pathways, AKT and ERK, are constitutively active in many cancers, and typically they're tied to phenotypes like cancer migration and other metastatic behaviors, which you can see on the right. So research efforts to control these different behaviors tend to focus on blocking one or both of these signaling pathways. But again, they're really intricately connected and not exclusive components that are tied to cancer behaviors. So what we're really hoping to do here is understand single cell responses in these pathways, how they're connected to each other, and how they fit into the larger signaling landscape. And we think that monitoring these dynamic signaling responses will help us to control and treat cancer. So central to the work that we'll talk a little bit more about today really is cancer cell signaling. So we'll touch more on some of the experimental approaches and modeling tools that we use to study these data. But ultimately, we're interested in looking at these signaling dynamics, linking that heterogeneity to precursors like differences in genetic inheritance, chemokine exposure, and environmental plasticity. And we also want to be able to model and use these signaling dynamics as sort of inputs to final cancer phenotypes, such as migration and proliferation and resistance. And ultimately answer questions like, why do cells respond differently to the same stimulus? And how do these responses change in different environments, for example? So ultimately, we have lots of pieces of information that we want to combine to understand sources of heterogeneity and inform mechanistic cancer treatment. And so now I'll hand it over to Patrick to talk more about our experimental pipeline and some of our preliminary modeling efforts.
Thanks, Ray. Okay, I'm controlling Ray's screen, so there's a little bit of a lag. So hopefully Ray convinced you that it's really important to measure signaling dynamics in individual cells so that we can understand heterogeneous behaviors in cancer. I want to give a brief rundown of how we do that. So the general flow is that we need to, in a specific cell line of interest, in our case, we use several different breast cancer cell lines, we need to express fluorescent reporters. Then we need to be able to do high throughput automated fluorescence microscopy to take fluorescence images of those cells. And it really helps in this process to have an automated image processing pipeline, which allows us to automatically extract data from these images. So I'm going to go through each of these steps in more detail. First, I want to talk about the reporters that we use. They're called kinase translocation reporters, or KTRs. The name stems from the fact that they are responsive to kinases, which are proteins that phosphorylate other proteins in the cell. AKT and ERK are both kinases. And the translocation refers to the fact that, as is shown in this diagram in the top right, when the kinase is activated, the fluorescent signal will move from the cytoplasm or from the nucleus to the cytoplasm. So on the left here, you've got a cell that has low ERK and most of the signal is in the nucleus. When ERK gets activated by, for instance, the addition of CXCL12, we see that signal move from the nucleus to the cytoplasm. One major benefit of this reporter strategy is that multiple reporters can be used in a single cell, since each reporter only takes up one fluorescent channel. So in our cells, we have the ERK KTR in the yellow channel, so it's attached to yellow fluorescent protein. And we have the AKT KTR attached to a cyan fluorescent protein, so that's in the blue channel. So in one cell, we're able to monitor ERK and AKT activity at the same time. So in this hypothetical cell, most of the yellow signal that we would see is in the nucleus, but most of the blue signal is in the cytoplasm, indicating that this individual cell has high AKT activity and low ERK activity at this particular time. We express the measurement of kinase activity using what we call the cytoplasmic to nuclear ratio, or the CNR. So to do that, we take the intensity of signal in the cytoplasm, divide it by the intensity of signal in the nucleus, and then we take the log base 2 of that, which centers that value around 0. We call that the CNR. So if you hear me say CNR, high means that the kinase is active, and low means that the kinase is inactive. So when we have these cells that are expressing fluorescent reporters, we next do high throughput microscopy on them. So we seed cells in 96 well plates. Each well might have a different cell type or a different condition. Then in each well, we can do time-lapse imaging before and after a stimulus is applied. So we would image cells in media. We might give them a different dose of CXCL12, or we might give them an inhibitor. The output of this process is a series of fluorescent time-lapse images. So for each well, we would have images of the cells in the AKT channel, the ERK channel, and also a nuclear marker channel, which is red. The nuclear marker is really important for tracking the cells over time, which we'll get to later. I also wanted to talk about the timeline of the experiment. So we tend to seed cells around 3 days prior, or 72 hours prior to imaging. Over this time, they grow. We have what we refer to as a precondition, which is just in this sort of relative time, around 4 hours before we give some stimulus to the cells, we might give them a different stimulus or an inhibitor in order to study how they respond to a change in their history. And then what we refer to as the 0 time is when we actually give the stimulus. So there's actually different timelines or different experimental timelines that we use for different questions. And so I sort of want to review that here. If we want to just measure heterogeneous responses to a ligand, for instance, maybe CXCL12, we would just image the cells right before we give a stimulus and right after. So maybe for 10 minutes before and maybe for an hour after. However, we might have a different question, which is, how does cellular history affect the response to a ligand? And we would measure that via preconditioning. So in this case, we might image the cells as we give a precondition, so maybe some inhibitor, and then we would measure the cell's response for 4 hours, and then we would give a stimulus and also measure the response to that. Now, a third experiment that is even a little bit more complex is, let's say we wanted to know about how sister cells behave. In that case, we would track cells for around 24 hours before a stimulus. This would allow us to see dividing cells and to know which cells are sisters when we give a stimulus. I do want to point out that as we move down and take more pictures from the top to the bottom, the experiments do grow in complexity. So it's not like we just want to take pictures of the cells sort of willy-nilly. We try to focus the imaging that we do to answer a specific question. But these are several questions, and sort of the goal of this is to give an intuition of some of the different choices we can make as experimentalists. So we do this fluorescence microscopy. What does it actually look like? So you can see a still frame of one picture. This is the ERK KTR in MCF7 breast cancer cells. So there's maybe a dozen cells on this slide. I've indicated 5 of them with arrows. The ones with red arrows, you can see, have mostly nuclear signals. So you can see this bright circle with a less bright haze around it. So these cells would have low ERK. However, there are a couple of other cells over here on the right and on the bottom indicated with blue arrows. And you'll notice that in those cells, you can't really see the nucleus stick out. So they have a more even ratio of cytoplasmic to nuclear signal. This indicates that these cells are more off, or they're more on compared to the cells where the nucleus is really bright. So we can already see, even in still shots, that we can start to see some heterogeneity in these cells. So on the next slide, I'm going to try a video. Not sure how well it will work for everybody. So I've also put still frames on the right. So I'm going to walk through the still frames first and then play the video a couple times. So in the top right, we've got those same cells. This is their initial signal. Again, you can see some cells are on, some cells are off. So there's some heterogeneity there. About 3 hours after this picture on the top right was taken, we added EGF to the cells. EGF is another ligand. It strongly activates the ERK and AKT pathways and is related to growth and will make cancer cells divide. So when we give EGF, we can see all of the signal go from the nucleus to the cytoplasm in almost every cell. So, these kind of look like donuts. We can see the nucleus is maybe darker in some cells than others, so there's still some heterogeneity there. But what's interesting is that if we look at the cells 2 hours after EGF is given, we can see this cell on the right still looks like a donut. Its nucleus is still sort of hollowed out. Meanwhile, the cell on the bottom left has a super bright nucleus. So this says that the cell in the top right is still really, really active in the ERK pathway 2 hours after EGF was applied, while this other cell has essentially entirely turned the ERK pathway off. This sort of naturally leads to questions about what causes the persistence of those signaling, of that signaling pathway. So I'm going to play the video and we'll see some other behaviors. I'm going to, okay, there it is. So I'm going to play this. I'll play it a few times. I would suggest just picking out a couple cells to look at and notice the flickering of the kinase activity and then the very concerted move of the signal from the nucleus into the cytoplasm as, I lost it, as EGF is applied. Okay, so you can see at the end, a lot of the cells are, or a lot of the signal is mostly in the cytoplasm. I'll play it again. And you can see some flickering too of, or some pulses of ERK activity as the cells are sort of just resting, they're sort of unperturbed. Okay, and so just to finish this out, so you can see right before the stimulus was applied, again, lots of the cells have nuclear signal. And if I just move forward a little bit, right after EGF is applied, you can see every cell has all of the signal in the cytoplasm, indicating that they are strongly active. So after we're done looking at these really cool videos, as I said, we have this automated image processing pipeline. So this pipeline was developed almost entirely by Kathy Luker, self-taught MATLAB expert. It's incredibly, I would say it's invaluable. It is necessary to do this work. So as I said, from these videos, or from this video microscopy, we get multiple fluorescent channels and we have time-lapse images. So the first step in the image processing pipeline is a process that we call nuclear masking. So we create a binary image from the nuclear channel that tells us where the nuclei are. Then we take those nuclei and we expand them a little bit. This approximates the shape of the cytoplasm. Remember, we need to get signal in the KTRs from both the nucleus and the cytoplasm in order to extract the kinase activity. So that tells us the kinase activity of individual cells at individual times. But remember, we're interested in the dynamics of these cells, so how they change over time. To answer that question, we need to connect the nuclei through time. So I've illustrated this process on the right. In the blue circles would be the nuclei from frame, let's say frame 10, and the red circles would be the nuclei from frame 11. And you can see the nuclei will generally overlap. And so we use that to track the cells. There's some other more complex methods that are based on probability in case there is no overlap. But generally, we're able to connect the nuclei through time in order to create dynamic tracks of kinase activity. So what does the data actually look like? Well, as I said in this cartoon, let's say we've got 3 cells. We can get their AKT and ERK activity over time in each individual cell. So this first cell maybe has relatively high AKT at the start, and then its AKT and ERK turn on over time. Maybe in the second cell, it's got lower AKT activity at the start, but it strongly activates AKT. Meanwhile, in a third cell, there's just no AKT activity at all. So this is just 3 cells. In reality, we are oftentimes able to get several hundred, if not several thousand cells in one experiment and in one condition. So it's not really feasible to look at just line graphs or scatter plots for thousands of cells. So we represent the data a little bit differently. So we do that using what we call color maps or chymographs. I've also heard them referred to as. They're kind of complicated, so I want to break them down step by step. On the y-axis of these color maps, we have individual cells. So going from 1 to 840 cells that were gathered in this experiment. So each row in this color map is an individual cell. Now along the x-axis, we have time going from 0 to around 250 minutes, and then going from 0 to 250 minutes again. The 2 columns refer to the AKT and ERK channels respectively on the left and the right. So what this means is that for an individual cell at an individual time, the color corresponds to its AKT or ERK activity, respectively. Low CNR or low AKT or ERK activity is dark blue and high is red. And so what this shows us is that there is heterogeneity in the starting state of both ERK and AKT. You can see that some cells have like sort of a yellow AKT value going all the way down to blue. And in ERK, cells are generally lower. Maybe the strongest cells are sort of light blue, and the most off cells are very dark blue. We can also see a concerted rise when we add EGF around 180 minutes. So that's over here on the right. But within this, we can also see some heterogeneity. So there's lots of different starting states for ERK and AKT. And also some cells activate and are really dark red, whereas other cells are, right here, they're just, they activate, but they only make it to sort of green or yellow. So there are clearly degrees of activation that are captured by these KTRs and represented in these color maps. So when we get all this data, the next question is sort of, what do you do with it? The first thing you might try is to just take the average. And that's not the worst thing to do. It certainly tells you some things, but I'm going to tell you it obscures signaling heterogeneity. So to show you how that kind of works, I've plotted the mean and the 25th and 75th percentile of AKT and ERK activity from the previous graph. So this does show us that AKT indeed starts higher than ERK in these cells. It also shows us that when you give EGF, you see a really strong rise in both AKT and ERK, and it's a bigger rise in ERK. It also maybe even tells us that the variance decreases. I don't know, the width between the 25th and 75th percentile maybe gets smaller after you add EGF. So certainly not no information, but it does raise a question. Do any cells actually behave like this black line? And the answer is generally no, very few cells do. So here I've superimposed 3 cells in green, red, and blue and their actual trajectory. And so when you look at these, you see strong fluctuations in both AKT and ERK. Some of that is for sure noise from the detector or from the image processing, but the image processing is pretty good. So some of these are actually real fluctuations in AKT and ERK behavior. And what this shows is that if we want to really understand the behaviors that these cells are exhibiting, we need to understand each cell individually. We can't just sort of lump them together and use sort of population-based statistics. Or if we do decide to do that, we'll be missing a lot of information. So one way we can do that is, for many bioinformatics people in here, this will make sense. If you have a high dimensional data set, a great way to make sense of it at first is to do dimensionality reduction. So that's what I'm going to talk about on this slide. There's one little wrinkle, and that is that Whereas something like a gene expression data set might just have, 100 or 1,000 genes in it with different observations. In that data set, the order that the genes are presented in doesn't really matter. However, in our data set, the data is fundamentally ordered in that there is time. You can't just swap the order of when the observations were obtained, or else you sort of destroy the time dependence in the data. So we use a technique called functional principal component analysis. It's an extension of PCA or principal component analysis to temporal data. And we applied PCA in this case to a data set where we stimulated cells with CXCL12. Now, one interesting thing about this data is we were able to segregate the cells based on whether they expressed the receptor CXCR4 or not. This is really important because if you don't express CXCR4, there's no way that the cells could sense CXCL12. So when we do FPCA on this data and we color the cells by whether they are X4 positive or X4 negative, we see two general populations of data. So up here on the left, this red cloud is CXCR4 negative, so no receptor. And we see this blue cloud stretching down and to the right. Those are the X4 positive cells. One thing that I do want to point out, though, is that lots of the blue cells overlap with the red cells. So just like in PCA, we can take all of these individual PCA scores and we can see, okay, what dynamics led to them. So when we do that, we see that the center of this blue cloud is a cell that strongly responds in the AKT channel, whereas the center of the red cloud is a cell that doesn't respond at all. But remember, lots of blue cells that do have the receptor are also in this red cloud, so they're also not responding. This has a sort of natural biological interpretation, which is that the receptor CXCR4 is necessary, but is not sufficient to signal. There must be something else in between CXCR4 and ERK and AKT that is making the cells not signal. So we can show the data again on a color map, and we can see these are the cells. In this case, they've been segregated by their X4 status. So all of the cells shown here have CXCR4. We can see some cells start green up here on the top left, and they strongly activate AKT going up to dark red. Other cells sort of down here in the bottom, they barely respond to CXCR4. So again, we see this observation that receptor is not, just because you have the receptor doesn't mean that you'll respond to CXCL12. So a previous student in the group, Phil Spinoza, built an ordinary differential equation model to understand why some cells don't respond and other cells strongly respond. So I'm not going to go deep into the equations in this model, but we can see that CXCL12 and CXCR4 receptor ligand interactions are represented up here on top. And the two species that we have KTRs for, AKT and ERK, are down here in the bottom. So each arrow on this plot is a differential equation representing an interaction between proteins. We can see that there's numerous species in between CXCR4 and ERK and AKT. Three of them that I want to call out are RAS, mTORC1, and PI3K. They have these little bubbles on them, and those indicate that those are sources of variation in the model. So Phil found that those three parameters are largely responsible for variation in AKT and ERK responsiveness. So what I mean by that is that Phil was able to simulate this model with several different parameter ranges for RAS, mTORC1, and PI3K. He did that, and since there were three parameters, it creates a three-dimensional space. So I'm showing that here. So the bottom shows the space in 3 dimensions, where mTORC1 is on the y-axis, PI3K is on the x-axis, and RAS sort of goes into the screen. And on top, I've got two different planes running through that surface. So on these planes, there's these blue and yellow contours. Those contours indicate regions of the model where there's strong AKT or ERK signaling, respectively, with strong AKT activity in blue and strong ERK activity in yellow. The gray contours indicate regions of the model where experimental cells actually exist. So if we go back to this example where we just had these two cells, one that strongly responds to AKT and the other one that doesn't, we can place them in this space. And we get these contours by placing not just 1 or 2 cells, but by placing around 1,000 cells. We can see sort of the contours of the population. So Phil was able to extract this cell state that was responsible for heterogeneity and signaling, and it allowed us to sort of map out where the cells exist. So on top, I have that same contour plot from the previous slide. On the bottom, I have an example of what happens when you precondition cells. So this is when you give FBS for 4 hours. FBS is fetal bovine serum. It contains a bunch of growth factors. You actually see cells move to a region of much higher responsiveness. Given that AKT and ERK are responsible for a bunch of oncogenic processes, we would imagine that cells that are down here are much more dangerous. So this sort of gives meaning to the heterogeneity that we see. So this is, at this point, I've sort of summarized some of the past work that we've done in the lab and some of the really interesting observations we were able to make from it. So Phil was able to model so remember we've got this timeline where we seed cells, 3 days prior, we give a precondition and then a stimulus. Phil was able to model the range of time right around the stimulus. So he was able to understand how heterogeneity, he was able to understand that cells are in heterogeneous states before a stimulus is applied, and that can cause a different response from one cell to another. However, there are other things that are not included in Phil's model. So for instance, in all of this time before the model, we're not explicitly modeling things like cell division or cell-cell communication, or perhaps even stochastic transitions between states, as maybe there's a fitness or a selective pressure for cells in the dish that causes them to move from one state to another. So clearly we have made some progress in understanding heterogeneous cell signaling, but there's lots of other things that we're really interested in understanding because we know that each of these things is responsible for generating subpopulations of cancer cells that are extremely dangerous and that we want to target pharmacologically in order to better treat cancer. So Ray is going to talk a little bit more about some of the future work that we've planned in the lab.
Yeah, so thanks, Patrick. So again, the ordinary differential equation model that Patrick just described is incredibly valuable to contextualize and place cells on that signaling landscape like the one shown here on the right. But one of the questions that we want to address specifically is just how that cell state is assigned. So for example, the 2 cells highlighted on this plot are in distinct regions of the landscape, and we can ask questions about what happened in the history of either of these 2 cells that caused them to land in these specific regions. Questions like, are they related to each other? And how close are they to each other in the dish? And are they receiving different cues from their environment and their cell neighbors? So broadly, we would like to understand how these cells behave collectively and over a longer time frame. And we have the tools to be able to do that. So just bringing up this timeline again, we have this ODE model to describe signaling right around the stimulus. But we want to be able to look way back in time and trace cells lineage-wise and how they're interacting with their neighbors and their environment. And one way that we can integrate all of these signaling data and lineage data and spatial information is by linking our experimental observations with agent-based modeling. And this agent-based modeling framework allows us to pose and understand mechanisms behind cell state assignment and generate hypotheses and make predictions about various populations of cells. So I'll talk a little bit more about the modeling in a bit, but first I wanted to show you some of the preliminary and ongoing evidence that we're gathering about cell relationships and signaling that we can use to inform our modeling. So really quickly, I want to bring your attention back to the image processing pipeline that we had discussed before. So Patrick mentioned how important it is for us to connect nuclei through time during the signaling part of our experiments so that we can trace KTR values and assign them to given cells and not just have a disjointed distribution of signaling values. But one addition that we needed to be aware of as we're beginning to image for longer periods of time and look for specific lineage relationships is connecting nuclei through cell divisions as well. So during the 4-hour time course of signaling, cells don't usually divide all that much. And even if they do, we tend to get rid of them from our analysis because the cytoplasmic to nuclear ratio gets a bit unreliable when the nuclear envelope disappears and a cell divides. And that's a fine decision to make in that specific scenario because we'll have fewer dividing cells that we need to throw away relative to the total number of cells in the population. But over 24 or 48 hours of imaging, we don't want to get rid of any cell that was a product of a cell division, because we would end up losing a lot of cells and not have relatedness information about them. So an additional piece that we implemented into this image processing pipeline, again, thanks largely to the efforts of Kathy Luker, was identifying characteristics of dividing cells and being able to link them through time. So when cells divide, their nuclei go through these very distinct and really cool morphological changes. So you'll see them shrink, get really, really bright as they duplicate all of their chromatin, and then abruptly produce two daughter cells that sort of push away from each other. So I'm showing schematically here in frame I+1, where the nuclei condense from blue to red, and then in I+2, where the cells separate into the green daughters. Before, we largely used overlap to connect cells through time, but now we've also implemented some probabilistic methods based on these characteristics of dividing cells to connect nearby cells and link them after a cell division. So next, I wanted to show you what this actually looks like in practice, mainly because looking at the cells is really exciting. So I'll let this GIF play for a couple of rounds, but I want to draw your attention to any one of the dividing cells. So you'll see, for example, this cell in the right has a really beautiful trajectory. It decides to move up the right side a little bit, and then it starts spinning, and all of a sudden, really, really quickly, we see it shrink, get bright, and then split into these two separate nuclei. So I'll let you guys watch this video again, and you can look at some specific cells or another dividing nucleus. But the point of this is just to show you some of the experimental intricacies that come into play when we're extracting lineage information from our microscopy images as cells are dividing. So now that we can track cells over longer periods of time and know how they're related to each other, be they sisters or cousins or what have you, we can begin looking at relationships between cells and understand how cell signaling behaviors are passed along family lines. So one key preliminary insight that we have into lineage influences on cell behavior is about the similarity of sister signaling. So looking at the response curves alone, we can pair groups of cells and calculate their squared residuals over the duration of their signaling. And an interesting observation that we make is that sisters are more highly correlated in their signaling in that they have lower residuals than pairs of random cells throughout the population. And if we're thinking about inheritance like a family tree, this is a logical and intuitive finding, but one that really sets the basis for us to be on the lookout for mechanisms that make sisters more alike than the rest of the population, and to be able to quantify this relationship and how it decays as we move through a cell lineage tree. And we can also take this one step further and look at the correlation of sister signaling over time. So if you'll recall, I mentioned genetic variation as one of the main drivers of heterogeneity in cancer. Now, in the previous slide, the data suggests that sisters are similar to each other. So for whatever reason, we're seeing this blue cell divide into two similar daughters that are also blue. Now, over the lifetime of these sister cells, you can imagine two different possible trajectories. One where that blue behavior is retained over the course of a lifetime, and one where that blue behavior transitions or decays and displays a lack of durability over time. And one thing that we can do here with the data we have is look at sister correlation over time to see if one of these trends, or to see trends and look and see if one of these durability mechanisms is more favored than the other. And what we see when we look at the data is that the sister cell coordination and signaling really remains consistent over time. So in this plot on the right, I'm showing you just how correlation varies with age. So we have old sisters on the left and younger sister pairs on the right. And the sum of the squared residual calculation between each pair is plotted on the y-axis. And the fact that we don't see a strong correlation between sister age and the residual calculation suggests that the first mechanism is at play. Otherwise, we might see younger sisters signal more similarly than older sisters, for example. And this opens up the floor for many more questions that we can ask, specifically about what factors are inherited that cause this similarity. Now, when a cell divides, it duplicates all of its contents, and certain proteins may be distributed evenly or differentially between the sisters. But we can also connect this back to the ordinary differential equation model and ask which specific signaling pathway components are responsible for this behavior. So if you'll recall, there are three values that really determine the cell signaling profile, and various combinations of inheritance between those three components might be responsible for the behaviors that we see. And so really understanding these types of questions about inheritance are questions that we have, along with a lot more. And we're really excited to explore these questions with experimental and modeling techniques down the road. And we're not only interested in understanding lineage relationships between cells, but also environmental impacts as well. So it's well documented in the literature that stromal cell interactions are really crucial in regulating cancer cell signaling. So stromal cells and cancer-associated fibroblasts, for example, secrete CXCL12 in the tumor microenvironment. So it's not a coincidence that we also stimulate our cells with CXCL12 to see them signal oncogenically. It's because CXCL12 tends to be a large component of the metastatic niche. And additionally, physical contact with stromal cells might force cells into having different behaviors, sort of in the way that at a crowded party, you might be at heightened alert as opposed to when you're alone in your room during the day. So if we go back to this cartoon that I've included with cancer cells in the center and stromal cells outside, we're hypothesizing that interactions between these groups of cells is going to be different based on where they're located physically. So you can imagine this inner region of blue cells that's completely shielded from the green stromal cells will behave differently than the red region of cells directly in contact with stromal cells. And again, this brings up a number of different questions like, what if there are several layers of cells? Or what if a single stromal cell gets inside? So these are the questions we have sort of thinking about in the back of our minds. But I want to show you how we go about approaching this experimentally. So in our experimental pipeline, we can co-culture cancer cells with stromal cells. And here you can see stromal cell nuclei highlighted in green and cancer cell nuclei either highlighted in blue or red. And we define these colors to represent cancer cells that are either exposed to stromal cells or protected from direct stromal cell contact according to some thresholds that we set up. And as you can see in this plot on the right, the mean signaling behavior for each of these groups, protected or exposed cancer cells, is different in both AKT in the blue and in ERK in the yellow and orange. And this again lets us begin thinking about teasing apart what exactly is different spatially and in these distinct neighborhoods that promote different signaling behaviors for these groups of cells, and how this ties into the genetic variability that we see between cancer cells alone. And this is where we want to be able to integrate all of these data streams again and bring some more modeling back in. So at this point, we have dynamic signaling data information, information about cell lineages, location history to look for things like migration, and also spatial information about neighbors, which presents a lot of different avenues to look into and a lot of different relationships that we can explore. So agent-based modeling is a really powerful modeling technique that we can use to integrate all of these data streams and really focus on how cancer cells acquire their signaling states and pass them along genetically, and also how these states are molded in various environments. So agent-based models are especially useful in these scenarios where individual interactions are important, when we care about the spatial organization of our system, and also when we have heterogeneous populations that can vary with some stochastic fluctuation. And most agent-based models are set up in a way where we have a number of agents. In this case, our agents are cancer cells, and we place them on a grid and allow them to perform some actions. So as we run the simulation through time, we monitor these 5 things on the right, namely the properties of the cancer cells, where they are, what they do, and how they decide to do it. More specifically, as we're looking to track the progression of cell state and interaction of cells through time, we're going to focus on this single cancer cell, for example, in the center that I've highlighted. Now, in the model, we were able to track properties like its location and lineage, age, and cell state. And these are all things that we can compare with experimental data over time. So these cells aren't stagnant, they perform many actions like dividing and migrating, and under the hood, updating their cell state. So in the agent-based model, we can assign rules to these behaviors that help us mechanistically understand why certain behaviors are occurring. So one example of this might be that a cancer cell only divides within a certain age range, or that in the presence of a stromal cell, a cancer cell updates any one of its signaling parameters by a certain amount. So now, based on each cell's individual history, trajectory, and interactions with its environment, we can interpret the signaling profiles that we see and explain why the highlighted center cancer cell doesn't behave like one of the other curves on this plot, for example. And we can use this framework to determine mechanistic rules that are consistent with experimental outputs, and also use this as a hypothesis generation tool to make predictions about ways to manipulate cancer cell state and test them experimentally. So I know we've been talking for a long time, but just to summarize some of the questions and tools that we're using, briefly, cancer is a heterogeneous single cell driven signaling disease. And we're looking at understanding heterogeneity in specific signaling pathways that are related to oncogenic cell behaviors. And we introduced some of the experimental tools that we're using to study cell signaling, namely time-lapse microscopy, multiplex kinase translocation reporters, and a custom image analysis pipeline that allows us to track cells and record their signaling profiles through time. We also talked about how we coupled these experiments with modeling both ordinary differential equations and agent-based models to interpret the experimental data and mechanistically explore the spatial and cell-to-cell signaling interactions. So I want to bring you back to this flowchart that I showed at the very beginning, where our goals are to integrate all of these different data streams, the inputs to cell signaling like genetic inheritance and drug exposure and microenvironmental effects, and also tie these to signaling outputs such as migration, proliferation, and resistance. And again, central to all of our work is the idea of cell signaling and modeling these behaviors. So I want to leave you with one final goal that we look towards. So if you'll recall, we're able to explain heterogeneity in cell behaviors on this signaling landscape with our ordinary differential equation model. Our broader, sort of longer term looking ahead goals are to be able to use this knowledge of cell state along with all of these inputs and outputs to target and manipulate cells in the signaling landscape to affect these outputs and treat cancer more effectively. So you can imagine mechanistically informed therapies to target specific subpopulations of cells based on signaling state, and also manipulations to push cells to more treatable or less dangerous regions of the signaling landscape. And with that, I'd like to end the talk and acknowledge all of the team members who make this work happen, because this is truly a collaborative experience with a number of different people. And with that, I think we'd be happy to field any questions.
I have a question. Thank you for the wonderful talk. These models are very detailed in their mechanisms, and it's great to see being able to connect the modeled mechanisms with the experimental data. I was curious if you could tell a little bit about where you're seeing the most computationally difficult parts of this. I can picture in some cases running the ODEs might be stiff and it's difficult to get them to converge, or perhaps fitting parameters of agent-based models can be challenging. So it's just, where are you spending most of your time in the lab, like person-wise and also computational-wise, where is the most CPU cycles spent?
I can take that, or at least Ray would love to hear your thoughts on ABMs. But one huge driver of computational complexity is in fact the image processing. So that is, it's definitely fast. Like it is. I would say it is as fast as we need it to be right now, because like my brain is only capable of taking in so much information, so it doesn't really matter if it can process the data instantly. But that is a really huge computational, that definitely takes a lot of CPU power. The ODEs are relatively, they are stiff, but they're, at least so far, we have been, it takes a manageable amount of time to run them. So when we generate like the signaling landscape, we run around 12,000 like combinations of those three species parameters. And I mean, that maybe takes 15 minutes. So there isn't a huge computational burden right now with the ODEs, I would say.
Right. And sort of piggybacking off of the ODE idea. So right now, I guess connecting our agent-based model to the ODE framework is sort of one region where we can expect some computational complexity. So right now, like Patrick mentioned, we sort of generate a library of signaling responses for a given stimulus or cell type, things like that. We can think about running the ODE model inside of the agent-based model sort of at every time step to understand signaling profiles through time, which may be a direction that we head in later on down the road. But right now, I think being able to sort of look at these cells through a lookup table is serving the purpose that we have, as we're not looking at like specific kinetic variations as cells are dividing and things like that.
Does anybody else have any other questions?
Yeah, and absolutely. If you're interested in reaching out, feel free to reach out to either Patrick or myself. We're always interested in discussing all of these aspects a little bit further with interested parties.
Yeah, I just realizing that was a shortcoming of the slides. I just put my email in the chat. So I love talking about this stuff. So yeah, please feel free to reach out if you have any questions or thoughts.
One more question if we have a few minutes.
Please.
You are looking primarily at breast cancer cell lines. And I was curious if you anticipated these mechanisms to be representable of cross cancers, or if you have plans of looking in other cell lines as well.
So there are a few ways to answer that question. We study breast cancer cells, or we study breast cancer. The Luker lab has a history of studying CXCR4, CXCL12 signaling, and that is a major driver of breast cancer metastasis, although it also affects other cell lines. So that's sort of the reason for that is a reason for the focus in breast cancer, amongst other, obviously it's incredibly clinically important. So generally, like the kinase networks that we study, ERK and AKT, are responsible for oncogenic signaling in just about any cancer you're interested in. So we generally expect that there are, like the signaling network that we described would be at least relevant in other cells. Although the caveat there is that since other cell lines would have other mutations and would have other microenvironmental inputs, for instance, different tumors would maybe, or like tumors in different parts of the body would be stiffer or softer, for instance, like glioblastoma in the brain is very soft. So we believe that like RAS is still going to activate ERK in every cell, but how that actually plays out in the cell is going to be then affected by other things. So we would expect that this idea that there is a pre-existing cell state that can control how cells respond to different ligands or different inputs. We think that that's a general idea, but I would hesitate to sort of extrapolate too far outside of breast cancer if we were to get to the point of making like very specific clinical recommendations. Does that kind of make sense? And yeah, Ray, feel free to add your thoughts.
Yeah, no, I agree with everything you said. I think the main takeaway is that this is like this is a platform that we're using. So I think different cell types, different genetic mutations, things like that, we can sort of plug and play once the system gets a bit more developed and say, okay, this is how breast cancer cells behave. Can we take and adapt our signaling landscape and understand behaviors in different cancers and make clinical recommendations based on the underlying behaviors that we're seeing from this framework. That makes sense.
This is really nice work. It's Kinnunen. And I appreciate your presenting it to our especially computational crowd, because we need to make more and more connections to the biological colleagues. I'm particularly interested about breast cancers, and I'm curious whether, of course, these are well-known pathways you've highlighted, but whether you've identified any differences specifically between estrogen progesterone receptor positive breast cancer, ERB2 breast cancer positive, and so-called triple negative of many different varieties. There's a lot of heterogeneity that's routinely identified in breast cancer studies. I just wonder if those stand out in your analyses also, or whether you've looked at it yet.
So I can say, so we've done a lot of comparisons. In Phil's first paper, he used MDA MB231 cells, which are triple negative. He used SUM159 cells, which I believe have estrogen receptor, although I'm a little bit fuzzy, and Veri cells, which are patient-derived cell line. We've been doing a lot of work in 231 cells and MCF7 cells. MCF7 cells do express estrogen receptor. So we're starting to sort of pick apart the differences in mutational backgrounds and sort of associated differences in cell lines. But we haven't, I guess I would say we haven't explicitly looked at like signaling from the estrogen receptor as an experimental variable, if that makes sense. It's more been, we've been comparing like cell lines with different mutational backgrounds. Does that start to answer your question? I'm not sure.
It's generally relevant, yes. On different mutational backgrounds, of course, one of the things that's a major resource, if you're not aware of, there's a lot of work contributed here from University of Michigan, is something called CPTAC. This is proteogenomics of 13 different kinds of cancers, including breast cancers. Here we've been working primarily on head and neck, lung adeno and lung squamous, pancreatic, and renal cell carcinoma. But there are others in the broad consortium of the NCI. What I was driving at was a little different. And we've had an interest going back a number of years on identifying splice isoforms of key proteins and transcripts in key pathways. And our focus was especially with the SUM149. I think that might be a pretty common cell line, and other lines with splice isoform analysis, both at the transcript and mass spectrometry-based proteomics level. And you can see important differences according to those finer details in the pathways. And if you have any way of getting such data for your analysis, I think would add some depth.
Yeah, that has been a major, that's been a major focus, is how do we integrate other streams of data? Because again, we sort of believe that the molecular interactions that we're modeling are general. PI3K is always going to activate AKT, but if a cell has more or less PI3K in terms of like transcripts, or maybe potentially, maybe it's localized in other places in the cell, those interactions are then going to be sort of modulated at a different scale. And that is something we need to take into account in our models, although it opens up other sort of difficulties with actually getting the data at a single cell level. But yeah, was that CPTAC?
Variation is from post-translational modifications of proteins. You cannot predict those from genes and transcripts.
Right.
World-leading analyst of those kinds of changes is right there in your department, Alexei Nezhvishky, in our department. Alexei Nezhvishky, he's also responsible for the Pathology Proteomics Service Laboratory, and he's a spectacular guy in the area of proteogenomics and computational aspects of proteomics.
Gotcha. Yeah, thank you for the heads up. And then did you say CPTAC? Was that CPTAC?
CPTAC, yeah, CPTAC,
Okay.
right.
Gotcha. I will check that out. Thank you very much.
Sure. Thank you again.
So I think we are at the hour, so I would just like to thank our speakers, and I think we can stop the recording.